Good evening, Arya. Okay. So I am Rahul Saha, and today we will be discussing the part 2 of the Krebs cycle, right? So we are discussing about the Krebs cycle, the tricarboxylic acid cycle, or also known as the citric acid cycle. And in the last class, we had talked about how the Krebs cycle gets broken down, right? In the sense that Krebs cycle, why does it even occur? It occurs due to the fact that you need to produce a lot of NADH, fat H2, lot of reducing equivalents. So therefore it is produced in that sense. So if we talk about Krebs cycle, okay, so in the last class we had discussed that Krebs cycle gets produced after oxaloacetate. It reacts with, you can say, your specific compound known as acetyl coenzyme A or acetyl CoA in order to produce citrate, right? And then in this specific reaction, oxaloacetate plus acetyl coenzyme A, what you have is citrate production. And this citrate is a very special molecule. While producing this specific acetyl, this citrate, the coenzyme A is released as COASH. Okay, thio group, it is released and H2O is taken in, and this citrate is the molecule because of which the naming of the cycle has been kept tricarboxylic acid cycle or the citric acid cycle. Citrate means citric acid, and this structure I had drawn it to you. I had drawn it for you in the last class. We have CH2 COO minus, then we have I can add OH. Then third one I can put again CH2 COO minus. How many carboxylic acid groups do you see? You see 3 carboxylic acid groups. That is tricarboxylic acid. Okay, that is why it is also known as tricarboxylic acid cycle. Okay. Good evening, Samish. Samish, just let me complete this one, then I'll start the errors in cell division. Okay. So I'll complete this thing and then we'll talk about the errors in the cell division cycle. Okay. Then the dehydration will happen, okay, leading to a double reaction known as, first by the enzyme known as aconitase. This aconitase causes dehydration and converts it into cis-acetate that we have talked about in the last class. Then again rehydration happens, okay. This aconitase causes rehydration again, means it will just, you can say, it will restructure itself, restructure the citrate, okay, realign it again and produce isocitrate. Now isocitrate, once isocitrate has been produced, now isocitrate contains, you can say, the 1, 2, 3, 4, 5 carbon molecules, right? And then this isocitrate is then converted into, you can say, alpha ketoglutarate. This isocitrate is then converted into alpha ketoglutarate. Okay, because 6 carbon molecules are present in case of citrate. One H2O goes. Okay, then after the, you can say, rehydration, again 6 carbon molecule is present in the isocitrate also. From 6 carbon molecule which is present in the isocitrate molecule, one CO2 will come out because it's an oxidative decarboxylation process. One CO2 will come out. In the last class I had shown it to you, and you get is 1 alpha ketoglutarate completely. And during this process, since this is a specific, you can say, oxidation process, something will be reduced. So NAD plus is reduced to produce NADH. So one NADH is produced from this specific reaction. Next is from alpha ketoglutarate, okay, that we'll be discussing in this class. alpha ketoglutarate is converted into, again, we will be saying that it is converted into, you can say, succinyl coenzyme A. How? What happens is you have specific alpha ketoglutarate dehydrogenase complex. That enzyme is dehydrogenase, which will go for the redox reaction, and in this case, the oxidation reaction. And then alpha ketoglutarate converts or gets converted into by this dehydrogenase enzyme, succinyl coenzyme A. Okay, it gets converted into succinyl coenzyme A, and succinyl coenzyme A. Okay, how does it form? Here one again COASH, that is coenzyme A group, comes in, okay, and then CO2 group is removed. Okay, one CO2 group gets removed. So what you have here is, you have, you can say, alpha ketoglutarate. If you draw, it was CH2 COO minus, okay, then CH2, then this side if you put C, put double O here, 1, 2, 3 bonds are done, both this, and then CO minus. So this is how alpha ketoglutarate looks. Here you can add CO2 oxidative decarboxylation, one more CO2 will come out. Here addition of coenzyme A, COASH group. Okay. Now, the succinyl coenzyme A, how will that look? Okay, structurally speaking, it will look like this. Again, CH2, CO minus, then CH2. Okay. Then you will be having C, here SCOA, okay, and then that CO minus group will go out. Okay, you will be having direct CO. So one CO2 went out. Okay, and this got attached with coenzyme A. This is succinyl coenzyme A. Now you can see one more thing clearly in biochemistry. First decarboxylation reaction occurred where? When pyruvate was oxidatively decarboxylated to form acetyl coenzyme A. Okay, via the pyruvate dehydrogenase complex, which contained E1, E2 and E3, right? This we had discussed in the last class only. So E1 is pyruvate dehydrogenase, this one is dihydrolipoyl transacetylase, this one is dihydrolipoyl dehydrogenase. Okay, three enzymes. It's a cluster of enzymes. You can say this pyruvate dehydrogenase is a huge enzymatic complex, cluster of three independent enzymes. And we also talked about how it required the function of thymine pyrophosphate lipase, coenzyme A, okay, then you can say FADH2. So all these were produced, okay. So NADH, okay, these were all required, okay, NAD plus. So all these things are required and this is being produced. So one CO2 was decarboxylated here, one CO2 was taken out or got released while producing alpha ketoglutarate from isocitrate. Here one CO2 came out, okay, and one CO2 came out while converting alpha ketoglutarate to succinyl coenzyme A. Okay, what does it mean now? It means that you have already taken out three CO2s, right? Three times decarboxylation has already been done completely. Now, three times decarboxylation has already been done means what? It means that three CO2s have been released. When three CO2s have been released means how many carbons did pyruvate contain? Pyruvic acid, three carbons. So the entire energy thermodynamically which was present in the pyruvate molecule, in the pyruvic acid molecule after it was converted from keto to enol form via, you can say, spontaneous tautomerization. So whatever energy was produced or was contained, whatever energy pyruvate was comprising has been converted or has been directly now used up. Okay, now the energy in the pyruvate has been completely used up, and now we can say that we have been able to produce CO2. Means we always talk about the fact biochemically that we exhale CO2. From where is CO2 produced? How does it get produced? It gets produced via oxidative decarboxylation reactions. That is how it does get produced. So you have one oxidative decarboxylation process going on in the conversion of pyruvate to acetyl coenzyme, another one from isocitrate to alpha ketoglutarate, another one is alpha ketoglutarate to succinyl coenzyme A. So we have had these kinds of reactions, and this will let the decarboxylation reaction happen, and CO2 is extracted out, CO2 is emitted, and that is why we are able to release CO2 via our breath. Okay, that is how we emit CO2, you can say. Okay, CO2 emanates from these specific, you can say, reactions. Now the next thing is, now we'll be coming on to the next step. What will happen to succinyl coenzyme A? Okay, now succinyl coenzyme A, before coming on to succinyl coenzyme A, okay, we have to understand some specific things here known as the thermodynamics. Okay, so when we talk about oxidation of isocitrate to alpha ketoglutarate, okay, what happens is, in this enzyme, the dehydrogenase enzyme, manganese is an important cofactor. Okay, manganese is needed in the active site. Okay, in order to activate this dehydrogenase enzyme. Okay, very important that fact is. Always remember Mn2 plus. So this, when this isocitrate is converted to alpha ketoglutarate, so this manganese is very essential. Okay, this Mn2 plus. Okay, very essential because it's the cofactor. And one more thing I'll tell you, there are two different forms of isocitrate dehydrogenase in all cells. One which requires NAD plus as electron acceptor, and the other which requires NADP plus as electron acceptor. Okay, the overall reactions are otherwise identical. Okay, in eukaryotic cell, the NAD plus dependent enzyme, okay, occurs in the mitochondrial matrix, okay, and serves in the citric acid cycle. Okay, and the NADP plus is present in both the mitochondrial matrix and the cytosol. Very important, this entire thing that I told you. In the human beings, especially eukaryotes, you can say, and humans also, because we do belong to the class eukaryota. Okay, so eukaryotes, what happens is, there are two classes of this isocitrate dehydrogenase enzyme that converts isocitrate to alpha ketoglutarate. Dehydrogen. One is class 1, uses NAD plus. Okay, here I told you that NAD plus will be converted to NADH. Right? Plus H plus. So it uses NAD plus. Okay, for the oxidation process, this needs to be reduced. Class 2 uses NADP plus, nicotinamide adenine dinucleotide phosphate. This is only dinucleotide. Okay, this is present in matrix, okay, of the mitochondria. This is present in both matrix and cytosol. Okay, So the dominant one regarding this Krebs cycle is class 1 molecule. Always remember this one. Okay. And the second one may be used in fatty acid and you can say sterol synthesis processes. Okay. Now what happens is you also have, okay, after this reaction you have oxidation of alpha ketoglutarate to succinyl CoA, right? Here again NAD plus serves as the electron acceptor, okay, and CoA is the serves as the carrier of the, you can say, succinyl group. Okay, so the thioester bond is formed. Thio means sulfur. SCOA that I told you, that bond is formed. Okay, it's a very spontaneous reaction. The del G for this reaction is minus 33.5. Del G naught at equilibrium for this reaction is minus 33.5 kilojoule per mole, you can say, for this reaction. After this you will be having alpha ketoglutarate to succinyl CoA. Same dehydrogenase, here alpha ketoglutarate dehydrogenase, okay. Oxidative decarboxylation CoA SH. Okay, so the del G naught is minus 33.5 kilojoule per mole. If you have studied thermodynamics, you will be knowing that negative del G means spontaneity in the reaction. Okay, negative del G gives free energy, and positive del S, that is the entropy, means energy is, the reaction is spontaneous. It will get proceeded in the forward direction. It will proceed in the forward direction towards the formation of the products. Okay, so it is very essential. So again the alpha ketoglutarate dehydrogenase complex here, okay, the enzyme alpha ketoglutarate dehydrogenase complex, it really closely resembles the pyruvate dehydrogenase complex, okay, in both structure and function. It also has three homologous enzymes associated with it, E1, E2 and E3, like the PDH complex, and also contains the enzyme bound thymine pyrophosphate, lipoate, then fat, flavin adenine dinucleotide, NAD, and coenzyme A. Okay, so they are, that means what? That both these enzymes, the application of this could be that both these complexes are certainly derived, you can say, from a common evolutionary ancestor, right? So that is the thing. So what happens is the complex, the, what to say, the even components of the two complexes are structurally similar, but what happens, that amino acid sequences differ. Okay, they have different binding specificities. Okay, the E2 components of the two complexes, okay, are also very similar. Okay, the subunits of E3 are really identical, you can say, in these two enzymes. Okay, so you can say that there has been some kind of divergent evolution which has led to them becoming alpha ketoglutarate dehydrogenase and pyruvate dehydrogenase complex becoming little bit different in their function because they catalyze different reactions, right? Now very important reaction, next one is conversion of, you can say, succinyl CoA to succinate. Very important next reaction. Now you have been able to produce the succinyl CoA. Now conversion of succinyl CoA to succinate. Okay. What I am doing is I have put a reversible arrow. Very important, remember. So what happens is the enzyme that is used, okay, is, you can say, succinyl CoA synthetase. Very important. First let's talk about the enzyme. Succinyl CoA synthetase. That is the enzyme that is used. Now what happens is the GDP plus PI becomes GTP and this CoA group comes out. Okay. One GTP is produced and this coenzyme A group comes out. If you see the structure, this CH2, CO minus, 4 carbon. O. So this was succinyl CoA. Okay. And if you see succinate, succinate will be 1, 2, 3, 4 carbons. So this will be your succinate. Okay. What happens is succinyl CoA. Okay. Like acetyl CoA has a thioester bond, right? The thioester bond has a very negative standard free energy of hydrolysis. This is del G naught. It is almost 30 minus 36. Okay. What happens is minus 36 kilojoule per mole. But what happens is in the next step of the reaction, energy released in the dissociation of this bond is used to drive the synthesis of a, you can say, phosphoanhydride bond, because we are phosphorylating either GTP or ATP. Okay, so the net GTP is only minus 2.9 kilojoule per mole. Okay, net del G of the reaction is only minus 2.9 kilojoule per mole. So I want you to understand it is not why, how it is 2.9 kilojoule per mole, because this bond when it breaks, it has a negative del G or del G naught standard free energy of minus 36. Okay, minus 36 kilojoule per mole. But what happens is this energy which has been derived is used to phosphorylate either GDP, and means add a pi group form GTP, else you can add a pi group to ADP and form ATP. So this is the phosphoanhydride bond, the last bond produced, okay, the gamma phosphate bond, and that bond creation takes a lot of energy. So what you see is almost 32 to 33 kilojoule of energy is spent there. So the net del G of the reaction after the formation of one GTP or one ATP is only minus 2.9 kilojoule per mole. Okay, so this is what you get as a result of that specific reaction. Okay, so succinate is formed in that process. Okay, that is why it is kind of reversible, because the del G naught is not that negative. Okay, so it can be a reversible process. So I have put, you can say, two arrows, that is the reversible arrows. Okay, so what happens is you can have a confusion here, because when I have produced the citrate, okay, oxaloacetate plus acetyl CoA, I use the enzyme citrate synthase. Okay, here I am using synthetase. Okay, difference between synthase and synthetases are, you can say, synthases catalyze condensation reactions in which no nucleoside triphosphate like ATP, GTP are used, okay, or required as an energy source. Whereas synthetases catalyze condensation reactions that do use, which do make use of ATP or any other nucleoside triphosphate as a source of energy for the reaction. So here GTP is being produced, you can say. Okay. So what you are seeing is here, that is why the enzyme is synthetase. Okay, always keep in mind that is why the enzyme is synthetase here. Okay, because GTP is being produced, so that energy is being used up to form GTP. Otherwise, GTP would have been used to drive this reaction in the, you can say, or any other reaction in the forward direction. That then again that would have been synthetase. Okay, like you have amino acid tRNA synthetases when we talk about in class 12, the tRNA charging. Okay, that requires one ATP molecule but requires two bonding breakage. One is the alpha bond, sorry, the gamma phosphate and the beta phosphate also. Okay, So the thing is, that is how the reaction goes on. Okay. So what happens is, now this is the reaction. Now what will happen is, you have produced succinate. Okay. Now you have been able to produce succinate. The next step will be oxidation of succinate to the molecule known as fumarate. Okay. Oxidation of succinate to the molecule known as fumarate or fumaric acid. Succinic acid to fumaric acid or succinate to fumarate. Okay. Now, when this reaction happens, this is again a reversible reaction. So I shall make it like this. Again, a reversible reaction. Okay. What happens is, you have got the succinate enzyme. Oh, sorry, succinate as the substrate, CH2. Okay. Then again CH2. Okay. So again you will be using the enzyme dehydrogenase. And then you will be using the enzyme succinate dehydrogenase, and here very special molecule will be used. We will not be using NAD plus, but we will be using as FAD as the electron acceptor. So FAD will get reduced to FADH2. Flavine adenine dinucleotide will become flavine adenine dinucleotide dihydrogen. FAD H2. Here NAD plus won't become NADH and H plus. FAD will be directly oxidized to, sorry, reduced to FADH2, and this is an oxidation process. So this has to be reduced. So flavine adenine dinucleotide is reduced to flavine adenine dinucleotide dihydrogen. Okay. And the energy of this reaction, okay, the entire standard free energy of this reaction del G naught is equal to 0 kilojoule per mole. 0 kilojoule per mole. That is why it is a reversible reaction. Okay, very important to know. It's a reversible reaction. So the entire mechanism containing the transfer of the phosphoryl group from the histidine residue, okay, to the terminal phosphate of GDP or ATP forming GTP. So those things are not there in your course. Okay, histidine 246 comes here. So I won't go into that detail. What happens is, in eukaryotes, just I'll tell you one thing, that the succinate dehydrogenase is not present in the matrix of mitochondria. Very important to know. Okay, mechanism, we will not go into the histidine 246 mechanism. Okay, nucleophilic attack and all. So we'll just stick to, in eukaryotes what happens is, the succinate dehydrogenase is an integral membrane protein of the mitochondrial inner membrane. Very important, you can note it down. This enzyme succinate dehydrogenase, okay, is an integral membrane protein of the mitochondrial inner membrane. Very important this is. Okay. So what happens is, in bacteria it is present as an integral membrane protein on the plasma membrane, but in eukaryotes it is present as the integral membrane protein of the mitochondrial inner membrane. It is also used extensively in complex 2 of the electron transport chain, which is also there in your syllabus, which we will be discussing very shortly. Okay, in the next few classes we will be dealing with that also, ETS. Okay, so the enzyme contains 3 different, you can say, specific clusters, which we will be talking about in detail in while ETS discussion, known as iron sulfur cluster, and one molecule of covalently bound FAD, flavine adenine dinucleotide. So iron sulfur cluster, just hold on for now. will be discussed properly when I'll be teaching you the electron transport chain or electron transport system. It is known by the acronym ETC or ETS. Okay. So electrons pass from the succinate through FAD and iron sulfur centers before entering the chain of electron carriers in the mitochondrial, you can say, inner membrane, or in case of the plasma membrane in case of bacteria. Okay. So this is how it is produced. This is how the reaction goes on. Okay. And how this entire reaction happens via histidine 246 residue pathway, that is not in your course, but this iron sulfur protein cluster, I'll explain it to you when I'll be explaining the electron transport chain in detail. That time we'll be discussing it in detail. Okay. And one more thing I'll tell you, one molecule is there known as malonate, which is an analog of succinate but not normally present in cells. It's a very strong competitive inhibitor of succinate dehydrogenase. Okay. And in laboratory dishes, or you can say laboratory cultures, in vitro culture, if it is added in the mitochondria or added to the mitochondria, to the cells, what happens is it completely blocks the citric acid cycle here, because then citrate cannot be converted to fumarate, because malonate has comparatively inhibited the citrate, sorry, this, then the succinate cannot be converted to, you can say, fumarate, because malonate has been able to competitively inhibit the succinate dehydrogenase enzyme, right, which is part of the, which is an integral membrane protein on the mitochondrial inner membrane. Next step is, in this cycle, we will convert fumarate to malate via hydration. So hydration of fumarate to malate. Next step. Fumarate to malate. What happens is, now we have fumarate. Okay. So I can draw it like this if you want to look at it in a better way. And then you can add the enzyme fumarase, and OH minus will be thrown out. Sorry, OH minus is added in. You are having hydration happening. OH minus is added in, the arrow should be opposite way, added in. Okay. Now what will happen is, you have a double bond here, you can break this double bond. Okay. So just look what I do. Just break this double bond, let it be like this. Okay, this double bond has been broken. So what I'll see is, I'll have, you can say, an OH group attached here. Okay. So here will be negative charge, here will be OH group attached. So this OH group, okay, is known as, this specific state is known as an carb anion transition state. Okay, you might have read these kinds of states in organic chemistry. Carb anion transition state. What will happen is, now H plus will come in the next step, again catalyzed by the fumarase. Okay. Here also fumarase will catalyze this. H plus. Okay. Now what will happen is, we will be able to add H here. Okay. So draw the same molecule. Here H, here H, you are adding here OH, then add one H here. So now this will be your, you can say, malate or L-malate, levomalate. Okay. So this is how the malate is produced. okay, via double reaction. This is also catalyzed by fumarase, this is also catalyzed by fumarase. Okay. The reaction is a two-step reaction. Okay, the hydration reaction, first OH group is added, then H plus group is added to the carbon ion. Okay, so that you have this minus group, then H plus group. So proper electrostatic attraction is present, and then you are able to produce this covalent bond. Okay, so this is the hydration process pertaining to the conversion of fumarate to malate. Okay, very important. The del G of this reaction is minus 3.8 kilojoule per mole. This is standard free energy, del G naught. Okay, it is not that negative, so I can make it reversible reaction, which I have done here. Okay, it is not highly electronegative. Sorry, it is not highly negative. So the reaction can occur in the backward direction too. Okay, so the enzyme is, you can say, is very highly stereospecific. Okay, it catalyzes the hydration. Here you should be seeing or observing is, that's why I drew it in that way, for you to understand that it catalyzes the hydration of the trans double bond of fumarate but not the cis double bond of malate. Okay, so in the reverse direction, okay, you can say from L malate to fumarase is okay, but D malate is not a substrate, you can say here. So you can see I have drawn a trans state here. This is the trans state. H this side, CO this side. So this is the substrate. Okay, trans fumarate. If I make it cis fumarate, it will not be a, you can say, substrate for this fumarase enzyme. Very important this one. Very stereo specific also it is. Okay, and then only L malate can be reversibly, you can say, catalyzed to produce, you can say, fumarate, but not D malate. Very stereo specific and also cis trans specific. Okay, this enzyme is. So our body's enzymes are really enthralling to watch how they can work. They will be able to recognize the stereospecificity, the L isomer, the D isomer, also the R and S isomerization, which is not in your course. Then also they can know which one is trans, which one is cis, the configurational state, okay, the configurational isomerism. So they can really, they are really a, you can say, spectacle in their own sense to behold, right? Now the next step is, you can say, oxidation of malate which has been produced, fumarate to malate. Okay, oxidation of malate is produced, is happens, oxidation of malate, and malate is converted to oxaloacetate. Okay, so succinate to fumarate, then fumarate to malate, then malate to oxaloacetate. So oxidation of malate to oxaloacetate. That is the next step. Okay, now oxidation means what? I'll draw L malate. Okay, L malate is OH, H, this side will be CO negative. Okay. This is our L malate. L malate, again reversible reaction. L malate dehydrogenase. Plus H plus. Okay. So since this is an oxidation reaction, NAD plus is again reduced to form NADH. and H plus. Okay, the third step. First one was when isocitrate was changed or catalyzed to alpha ketoglutarate via isocitrate dehydrogenase. Okay, class 1 mostly. So it used NAD plus as the cofactor. Okay, NAD plus was reduced to NADH. Then the alpha ketoglutarate was changed or was again oxidatively decarboxylated to succinyl coenzyme A. Okay, that happened. Then you had succinyl coenzyme A becoming succinate. Now again succinate. Okay, it became from succinate to fumarate, right? So that reaction also had this specific, you can say, FAD as the cofactor, and FAD H2 was produced, you can say. Okay, so succinate to fumarate. So 2 NADH has already been, so 2 NADH molecules have already been produced, one FAD H2 has been produced, and now this third NADH molecule is being produced in the oxidation reaction of, you can say, this last molecule is your malate to oxaloacetate. Okay, very very important, L malate to oxaloacetate. So NAD plus becoming NADH and H plus. So how does an acetyl and oxaloacetate look? Okay, it will be CO, we have CO minus, okay, then CH2, CO minus. So this is how an oxaloacetate looks. Okay. So what happens is the del G of this reaction, understand very very carefully, the del G of this reaction is, or the standard free energy del G naught of this reaction is 29.7 kilojoule per mole plus 29.7 kilojoule per mole. Now the question you would be asking me thermodynamically is, how is this reaction possible in the cells then? Because this is a completely non-spontaneous reaction, because del G is heavily positive here, not 1 or 2 that I can make it reversible somehow. So how does this reaction occur in the physiological pH inside the cells? pH of 7.35 to 7.45 in the, you can say, under the physiological conditions in the eukaryotic cells, and this reaction does happen, otherwise you will have no concentration, no formation of oxaloacetate, and thereby Krebs cycle will be ceased. How does it occur? So the equilibrium of this reaction, we know that it lies far to the left under standard thermodynamic conditions, means it will favor the formation of reactant, not the product, not to the right hand side. But in intact cells, what happens is the oxaloacetate produced is continually removed by the highly exergonic citrate synthase reaction, which is the step 1 of the Krebs cycle. Okay, you can see here. So what happens is this keeps the concentration of oxaloacetate in the cell extremely low, below 10 to the power minus 6 molar. Always remember, below 10 to the power minus 6 molar. Okay, molarity. Thus, it pulls the malate dehydrogenase reaction toward the formation of oxaloacetate. So that is the entire theory behind it being able to produce oxaloacetate in the first place. Okay, it is continuously removed, and how it is removed? Because the citrate synthase reaction has a very very negative del G. Okay, so exergonic reaction means exothermic reactions, energy is liberated, and oxaloacetate is not present in the large amount in the or on the product side. So thus it can be able to proceed in the or towards the formation of the product. Okay, even though the del G is quite highly positive here. So this is how the reaction continues. Okay, so the entire thing is, if you look at the Krebs cycle, so now this entire Krebs cycle is completed. We have 3 NADH produced, We have one fad H2 produced. These three NADH and one fad H2 produced are the core treasures why this entire Krebs cycle, TCA cycle of citric acid was initiated or commenced in the first place. It didn't commence just to produce one GTP/one ATP molecule. Okay, such meager process would not be continued by the body just to produce one ATP or GTP. When in glycolysis, two ATPs produced more than this, or are produced which are more than this one GTP/ATP. But the thing is here, four reducing equivalents are produced per Krebs cycle. But you have two pyruvate molecules, if you remember, after the glycolysis has got over. So they'll enter the mitochondrial inner membrane, okay, via pyruvate mitochondrial pyruvate carrier MPC protein, and again one Krebs cycle, one round of Krebs cycle will occur. So two pyruvates which are produced, which are synthesized after or as a consequence of glycolysis, they will yield how many Krebs cycles? Two Krebs cycles means they will yield two GTP/ATP and eight reducing equivalents. So one glucose molecule results into formation of 8 reducing equivalents, 6 NADH and 2 FADH2, which will be proving to be, or which will prove themselves to be very, very pivotal for the initiation of the electron transport chain. Otherwise, electron transport chain would never begin and will never be able to produce almost 30 ATP, okay, through the F1 F0 ATPase as a result of one glucose molecule breakdown via glycolysis. So this is the end result, or this is why this entire reaction goes on, okay, in the forward direction, and which is known by the name as Krebs cycle. Okay, very, very important. So I'll just show you how the Krebs cycle looks. Okay, it looks like a circle, okay, because it keeps on returning to the same stage. Okay, Krebs cycle. Let's see here. So it looks like this. Let's zoom in the image. Okay. So it does look like this because it returns here. This will again citrate synthase COSH. It will again form, you can say, isocitrate, cis aconitate, then isocitrate will be again produced. Okay. So this reaction keeps on happening, and this is the citric acid cycle or Krebs cycle or tricarboxylic acid cycle. So I hope you are able to comprehend all the concepts which have been taught today. If you have had any queries or doubt, you can contact me on my personal number. Okay, from next class we will be beginning the errors in this cell division. Okay, then we will be returning back to the biological transformations because we have a lot of cycles remaining. ETS is remaining, F1 F0 ATPase is remaining, C3 cycle, C4 cycle, those are remaining too. Okay then, so let's meet you in the next class. Thanks a lot, and let's meet you in the next biotechnology class.